these are my disclosures. It's an honor to be here, and I wanted to make sure that everybody was clear of all my relationships. If anything is a conflict, I will definitely announce it during the presentation. The objectives today, I wanted to review again current ALS treatments and review the importance of multidisciplinary care. But I also wanted to highlight the clinical trial pipeline in the US. I'll also highlight some things that are occurring outside of the US. And then talk a little bit about pre-approval access to investigational drugs outside of a clinical trial. And then really the heart of the presentation is talking about challenges and opportunities, which complement a lot of what we've discussed together as a group today during this Clinical Research Learning Institute. And I want to thank again the ALS Hope Foundation for allowing this get together today. So first, the current treatment options for ALS. As many of you know, the two FDA-approved drugs are Riluzole and Edaravone. Edaravone's brand name is Radicava. So people ask me, you know, Riluzole, when I see someone who is first diagnosed with ALS, they ask me, okay, Riluzole, is it going to help my function? Is it going to help my breathing? What is the data that helped it get approved? And so Riluzole was approved in the US in the 1990s, and it was based on 2 clinical trials. And essentially it had a survival benefit. And the benefit on average between the 2 trials was 2 to 3 months. And so that's what people hear when they talk about Riluzole. But if you look at more recent publications, there was one study that looked at staging out of the UK, right? And it unfortunately, the people that entered the clinical trial were beyond stage 1, which is the earliest stage for King staging, but it did show some delay of progression or delay of progression from stage 4 to 5, which is a later stage. But again, you couldn't really assess the earlier stage transitions because patients were too advanced. Another study looked at population-based databases, and these looked at median survival times and average survival times in different countries post-approval. So this is like an attempt at looking at real-world data. And in the clinical trials, often people will discuss this 2 to 3 month advantage over placebo in the clinical trials, but in those real-world databases globally, the median survival benefit if people were on Riluzole was about 6 months to about 19 months. So it's a little bit better than what the clinical trial did. And of course, there are other factors that are unanswered, like if you started earlier, is there a bigger benefit? These are not controlled studies. And so there's a lot of other confounding variables with regions and care and other medications. But I just put up here what the dosing should be. There's an oral tablet that was originally approved. Then the oral liquid form came more recently in 2018, and a more recent formulation of the sublingual oral film. The second medicine that came, so we went a long time and we did a lot of clinical trials. We were really excited after Riluzole, and we really hoped for a drug to be approved just after that, but it wasn't until 2017 that Edaravone or Radicava intravenously gets approved in the US. And that was based out of a 6-month study out of Japan and 137 patients that showed a difference between the ALS functional rating scale. Now, I showed you the data from Riluzole that preceded the development of ALS functional rating scale, which we commonly use for outcome measures in clinical trials and also assess function in clinic. So this study showed a difference of 2.49 in between the treated group and the placebo group. And based on that study, an additional supportive study from the open label extension, it was approved in the US. And the dosing is as follows. It's actually a complicated prescription to write, but there's cycles, and the cycles for the first cycle, it's considered the loading dose where you do 14 days of intravenous dosing. It's 1 hour through any kind of venous access. So either PICC line or a port catheter, or sometimes people might initiate with just a peripheral IV, but that's not a long-term solution. And then subsequent cycles, people will get 10 days in a 14-day period. So for example, if you look at 2 weeks, you can get dosed Monday, Tuesday, Wednesday, Thursday, Friday, take a break Saturday, Sunday, and then get Monday through Friday dosing the second week. And as some of us talked about earlier, and which is why I was scrambling to redo my slides, yesterday, the FDA approved the oral formulation or the oral suspension of Edaravone. I think one of the major challenges personally for me as a physician when I treat people living with ALS, it's been a challenge to manage the peripheral IV access long-term. Have struggled with people being fearful of the IV access, or struggling with trying to care for it because the PICC lines dangle out of the arm and they get caught in clothes. I'm in New York, so wintertime, it's always an issue. Sometimes they malfunction or sometimes they get infected. So the oral form is theoretically could be easier. The dosing, interestingly, is the same. It's still that loading dose of 14 days and then 10 days each subsequent cycle. And then on the horizon, there was already, many of you are already aware of AMX0035 being developed by Amylyx. It already underwent an advisory committee meeting. Just to be clear, an advisory committee is an independent panel, and they review the testimony and the data provided to them to make their determinations, but that's not binding to the FDA. So that's just a recommendation. So the FDA will take their input as well as all the other inputs that they have in their own analyses to make their independent decision. And that date is June 29th. So it can occur anytime before or at June 29th. So we're following that story closely. And then this week on my birthday, the Japanese published a subsequent study, a second study actually, on ultra high dose methylcobalamin, which was done in 130 patients, and the observation period was 16 weeks. And in that, and I hope the windows are not blocking the window is not being blocked, but essentially the study showed a difference of 1.97 over 16 weeks. That's 4 months, so shorter than our typical 6-month studies. And it was statistically significant. And so this actually is something in conjunction with the previous study with ultra high dose methylcobalamin. So the dosing for that was 50. So methylcobalamin is B12, but it's not your typical formulation that you use for vitamin B12 deficiency. So that formulation is usually called cyanocobalamin. So you got to excuse the way I say it. Methylcobalamin is a different formulation, and the way it was dosed in this study is 50 milligrams injected intramuscularly twice a week. So it's two injections twice a week, like Tuesday, Thursday, or Monday and Friday. And so that came out as well. And there is some discussions, and maybe it would be another thing that would be added into our treatment regimen, or may proceed to possibly review and regulatory review for an indication in ALS. The only thing I'll say here is everyone is familiar with multidisciplinary care. Why it's so important, I just want to clarify, because every time I see somebody who's newly diagnosed, they ask me for a drug. What can I take? What can I take? Okay, so you only have riluzole and radicava. What else can I take to help my disease? And I often take a minute to step back and emphasize that a lot of experimental therapies or investigational products, we don't know whether they help. And it's important to understand that multidisciplinary care, so that means that there are many healthcare providers during your visit with the neurologist. So there's a physical therapist, an occupational therapist, speech, social work, neuropsychology, GI, pulmonary, and they all kind of help all of the things that are affected in ALS. And that type of care does help to improve survival and quality of life. The other aspects of that multidisciplinary care include dietary and nutritional management and respiratory management in a very proactive manner. And the reason why that's important is that it can modify disease progression and improve quality of life as well. And so that's why pulmonary care and dietitians and managing your weight and monitoring your weight becomes so important as part of your care. And some of the data and evidence for that is better than anything else in terms of experimental therapy that we have right now. And with those items, plus the FDA approved medications, I always focus on that as the first step before we start talking about other things down the line. So then we get to the current clinical trial landscape. I'm focusing on the US because we're US based. But of course, all of us and all the clinicians that are involved in the CRLI are involved globally with researchers. But when should a clinical trial be considered? The clinical trial, you should discuss it. Obviously trying to get involved in it as early as possible is always important, but it's important to have already discussed and reviewed the risks and benefits of the FDA approved treatments with your healthcare team. You may choose to take it, you may choose not to, based on that information, and that's your choice, but it's also important to know that these should be reviewed as early as possible. The second point is that you're actively being cared at an ALS multidisciplinary clinic. And I explained why that's important. And the last is that you've discussed and reviewed whether you need genetic testing. And why is that important? Because if you're interested in a clinical trial, there are clinical trials for people who have no genetic mutations associated with their disease. But if you do have a genetic mutation that's associated with your ALS, you might be eligible for a specific trial targeting that particular gene mutation. Now, one thing to remember is as you're going down the algorithm is that many clinical trials will allow you to be on a stable dose of an FDA-approved treatment for ALS. So for example, Riluzole is often allowed if you've been on it for 30 days or longer and everything's been stable and you're tolerating it. You may be on Riluzole or Radicava for at least 1 or 2 cycles, and that'll be okay. But once you enter a clinical trial, you're not allowed to start those things in the middle of a clinical trial. So it's really important to make those decisions, or at least try them before you jump into a clinical trial. Once you're in a clinical trial, and this is a loophole I always talk about, but I don't think a lot of people know, is once you're in a clinical trial and you decide that maybe the Riluzole is not effective, or it might be causing a new side effect, or the Radicava might be causing a side effect, and you need to stop it, you're totally allowed to stop those medications and then continue on the clinical trial experimental therapy. So it's really important to have those discussions about the approved therapies first before starting into a clinical trial. Now, when we talk about clinical trials, we've talked a little bit about this, but just to make the point, there's interventional clinical trials that test a potential therapy. It could be a device even, and participants can be assigned to either a drug, investigational drug, or a placebo, or it could be just a control arm. And it's used to determine the effectiveness of a potential intervention. So whether that's a drug or a device. Observational studies are when you're enrolled and there's no treatment given, but we learn about the disease itself, you know, how a blood marker might change over time, or what might be the significant signal, and try to understand people's individual diseases. And this is often how we develop a biomarker for disease progression, or a biomarker as a diagnostic test, or understanding the differences in populations. Now, I'll focus on clinical trials because a lot of people are going to give talks during this meeting that touch on a lot of these points. But right now, therapeutic approaches or clinical trials evaluating investigational therapies focus on several issues. One, like I mentioned before, is targeting specific gene mutations. So since there are clinical trials targeting specific genes, and not every gene, because you've seen a slide where there was lots and lots of genes, and every year we seem to have exponential growth of the number of genes we're aware of, but it's pressuring us as a clinical community to ensure that people have access to genetic testing and that we identify them as early as possible, and try to have a way of communicating, and that we all talk the same way, about what the results are and how to handle things like variants of unknown significance and so on. So it's going to be important to incorporate genetic counseling along with the testing. Most of the drugs that are in the pipeline for ALS target various pathways that are affected in motor neuron degeneration. So I just listed just a few. I have to say most of the drugs that are in early phase 2 or phase 3 are targeting neuroinflammation, so complement inhibition and kinase inhibition. Some drugs help to improve how the nerve and muscle communicate, and then there are some drugs that are targeting specifically the muscle themselves to help improve contraction, the strength of the contraction, and reduce fatigability. And of course, there's cell therapy and stem cell investigational trials, which I'll touch on just briefly. So this is just a snapshot to kind of give you a sense of where things are in the US. So as you can see, we're tilted a little bit towards early phase development. The HERV-K phase 1 study is out of the NIH. This is looking at, and actually it's a way of kind of doing precision type experimental clinical trials. So they're looking at people who have elevated HERV-K in their blood, and then using this therapy to try to see if they can lower it and have a clinical effect. BLZ 945 is being developed by Novartis, and it's a CSF inhibitor, and it essentially reduces the macrophage or neuroinflammation that occurs, but it's a phase 1, so it's a short dosing study. Many of these on the list are targeting neuroinflammation, so complement inhibition, and some are kinase inhibitors like the RIPK1, which is now recruiting. That's the Himalaya study. And WP0512 is a Rho kinase inhibitor that also goes by Fasudil. So that's also, those are newer ones that are on the list. There's the platform trial, which many of you are familiar with, and regimen E is Trehalose, which is regimen E, which has initiated enrollment as well. And then the phase 3, we have Abutulast, Masitinib, which targets some forms. So Masitinib is a tyrosine kinase inhibitor, and Ibutulast does target some neuroinflammatory pathways. And Reldesemtiv is a skeletal muscle activator in phase 3, and it's in a study called Courage ALS. And then I added the oral edaravone for approved therapies. I put Nudexta here. Nudexta's FDA label indication is for helping with easy laughing and crying, what we call pseudobulbar affect. And it's really effective for people who experience that symptom. But there was a subsequent study after its approval that was published in Neurotherapeutics, I believe in 2017 by Richard Smith, that showed in a 3-month study that people with ALS, if they have bulbar symptoms or speaking or swallowing issues, and they take Nudexta, they had perceived either plateauing or improvement of those symptoms. And so if someone has speaking or swallowing difficulty, often there's a discussion about the risks and benefits of Nudexta for that subset of people living with ALS. Now, the gene therapy list keeps growing here. And so first I'll talk about cell therapy. Cell therapy does not mean stem cell therapy. And so T regulatory cells therapy is one type of cell therapy. There's autologous or derived from your own body T regulatory cell therapy. So often the T reg cells are taken out of your body. They're taught to behave. I mean, I'm going to oversimplify this, so my colleagues don't, feel free to embarrass me. But it's essentially to teach those T regs to behave better and then put them back in your body. So there's 2 companies that are working on that type of therapy, which is Calico and Rappa Therapeutics, and that's actively recruiting an early phase. And then there will be another form of what's called allogenic T cell therapy. And that's deriving those T reg cells instead of from your own body, from cord blood. And they have never been exposed and they're naive, and hopefully they'll behave better. And so those are being infused. Those are planned to be infused in early phase clinical trials. And the company that's developing that is Solenkos. I didn't put a star next to CNS10 NPC GDNF. So those are human neural progenitor cells. Cedars-Sinai in California is actually recruiting for a phase 1 study where they're going to do direct injections into the brain in a phase 1 clinical trial. So they are looking for early stage volunteers that would like to participate in that study. And then the Mayo clinics are also recruiting for a phase 2 adipose-derived mesenchymal stem cell study as well. This is where they take fat cells from the belly or the abdomen, turn them into stem cells, and then infuse it into the spinal fluid. And then the gene-directed therapies or specific therapies targeting a specific gene mutation has grown, actually. So many of you are already familiar with the Tofersen program, which is now only available through an early access program. But recently, a smaller phase 2 study using monoclonal antibodies targeting the SOD1 protein called AP101 has been launched globally, actually. Then also several compounds targeting C9orf72 are also actively recruiting, and it's amazing because that list keeps growing. And then ataxin-2 and FUS are the other genes that are important to screen for or know about in your own body so that you know if you're available or you're eligible to participate in that clinical trial. So what are some things? So I think one of the biggest questions I get from people is, okay, thanks, Dr. Andrews, that was a lot of information and I'm quite overwhelmed. And you just told me that I might be eligible for maybe 4 clinical trials or 1 clinical trial. And I know that's not the case for everybody. But for those that would be eligible, it becomes difficult for them to decide which one would be ideal for them. And so it's important when you're looking, whether you're eligible or not, it's when you're looking at any potential clinical trials or experimental therapies, you want to look at what phase of development that drug is in. What is the mechanism of action for that drug? And, is it targeting the motor neuron, a pathway in the motor neuron? Is it the muscle? And what is important to you? Are there any medical contraindications? For example, if there's a drug that elevates your sugars and you have diabetes, that might not be the ideal drug for you. What are the known side effects? So as you move through the phases of development, you learn a lot about side effects. So the earlier you are in phase, so phase 1 is usually first in human, you don't know a lot about the side effects. But as you move to phase 2 and phase 3 of clinical trials, and you get later in the clinical development program, you'll learn a little bit more. But you still, I emphasize to people who enroll in phase 3 clinical trials, people feel confident, but we still don't know everything about that drug and how it would affect a lot of people. So there are still unknowns even in phase 3. An important question nowadays is, how is the drug administered? How do I take this drug? Do I have to come into the hospital for the drug to be administered? Or is it IV? And does that mean I have to make frequent trips to the hospital, or can I get that IV done at home? Is it orally taken? Can I take it through my PEG tube or my feeding tube? These are important questions to ask. And then for many people, it's important for them to understand if there's a placebo arm. So what are my chances of placebo? Dr. Ilieva just spoke about informed consent. In your informed consent, there's always a discussion about your chances of receiving treatment versus placebo. And you should really, if you don't see it, you should ask whoever's obtaining informed consent to go over that with you and look at the chances of treatment versus placebo. Many trials now, if there is placebo, they will make it so that it's the randomization is tilted towards favoring your chances of being on treatment. Or if there is a placebo, often the people who are designing it will try to make it as short as possible so that after you're completed, you can roll over to what's called an open label extension, where you're guaranteed access to the drug after a commitment of time where you may be on placebo. And then asking about how frequent the visits are and how far the location is and can you get there. So say you've gone through the FDA approved medications, you are connected with a multidisciplinary clinic, you've reviewed all the clinical trials, but those are not options for you. So what do you do then? So the time that you start having the conversation is after you've talked about the approved therapies, you're in an ALS center and you've discussed genetic testing and you've looked at all the potential clinical trials. There's nothing right now for you. And you've considered even alternative or potentially off-label use after you've talked about the risks and benefits with your provider and reviewed the rationale for considering this access for investigational therapy outside of a clinical trial. So pre-approval access means that you are asking for a drug that is not yet FDA approved for ALS. So the most common way people get it is through a clinical trial. Sometimes people will get what's called off-label medications or drugs. So this is when a drug, technically it is FDA approved, but it's not approved for the condition that it's being prescribed for. So that's called off-label. That's different than getting something over the counter. So there's a lot of supplements that have good rationale for helping the motor neuron survive. Those can be purchased over the counter, different, and don't require a prescription. Off-label means you're writing the prescription or you're getting a prescription from a physician. Then outside of a clinical trial, outside of off-label, there's expanded access. And that program has been in existence for a while. And sometimes it's referred to as compassionate use. Abroad, the program is called compassionate use, but in the US, it's called expanded access formally. And it provides a pathway for people to gain access. More recently, there was legislation that passed for right to try, but despite the law being passed, there hasn't been many opportunities to obtain drugs through that pathway, especially for ALS. I actually don't know of any except for one case since its approval. And so expanded access is used more frequently, and FDA has approved access for ALS historically. So it is really, originally it was intended as a treatment. So it's not a clinical trial where we're trying to learn about the drug and the safety and tolerability to lead to its approval. Expanded access was just for people who were in a serious life-threatening situation and all other options were exhausted. And there are different types of expanded access programs. So there's ones that you can apply for an individual. So for this, a physician and a person living with ALS will come to a physician, will ask about the drug. They will make a joint request to a company who might be manufacturing that drug. They have to get permission, and they have to be allowed access to it from the drug company. And then there needs to be an application that's put in on the behalf of the patient to the FDA that would need to be approved. And subsequent to that, the physician would then have to apply to their local ethics committee or institutional review board, have that approved, and there's a formal informed consent. And once the drug arrives to the site, then the patient gains access. Typically in that scenario, it's called a non-emergency use. In an emergency use is when in a life-threatening situation, a person can't provide consent, the healthcare team can't actually wait the time for an FDA approval and may use a drug. So sometimes this has been used in COVID. And then after it's been used, they will ask for the approvals. And that's a really rare instance. In intermediate size populations, this is where 15, 20, there's no specific size listed, but it's for a number of patients populations. So you would apply to treat X number of people with a specific investigational drug. And a treatment protocol is typically sponsored by a pharmaceutical company. So it's like when you have a global phase 3 study and you have a whole lot of thousands of patients that want access after the trial, and they might allow for an expanded access. And so they would sponsor it and have it in multiple sites, multiple regions. So the criteria is if someone has serious or immediately life-threatening condition, and most importantly, in the legislation, it says that it should not interfere with initiation, conduct, or completion of a clinical trial, especially for that drug. And the submission can be made by a physician, or who would be considered the sponsor, or it could be made by the drug company. I just put this slide here because, of course, EAPs or expanded access has been a growing topic of importance in the ALS community. And as a healthcare provider and as a physician in a clinic, I've experienced some challenges in trying to do this for people who request it. So first and foremost, a drug has to be made available by the drug manufacturer, and they need to have adequate supply. Many of the companies that are in the ALS space are small or have limited resources, and it's difficult for them because they have to also complete the clinical trial they may have already initiated. But if me and whoever I'm treating want to get access, we have to get access from the drug company first. And like I mentioned, a site has to complete the FDA application and receive approval. And there's no dedicated team, there's no dedicated time, got to find time to do it and have to do it. And then on top of maintaining our clinics and maintaining our clinical trials, and often we'll pull resources from our clinical trials in order to get the paperwork into the FDA and also to our IRB. But once we get all those done, and if the drug company says they'll give me the drug, sometimes these drugs require specific storage, and that cost may not be covered. Insurance doesn't cover it, hospital doesn't want to cover it, and unless there's philanthropic dollars or other kind of excess funds that can be used, the costs are not covered. And then if the investigational drug has to be administered at a hospital, so for example, anything that requires a spinal tap and in the hospital and administered into the spinal fluid, for example, or a procedure, we require a hospital facility and procedure, and I might have to rely on another service to do the procedure, and those costs may not be covered, and the hospital may not let me use the space for that particular reason. So there's sometimes these technical issues that kind of get in the way. If the investigational drug can be taken at home, it's easier, but sometimes the drug is shipped to the site and then we have to ship it to the person, and then sometimes that cost is out of pocket for the person who's receiving the drug. And then even though it's not a clinical trial, I'm still subject to restrictions that are common in a clinical trial. So for example, if I give an investigational drug to somebody, I am obligated to monitor their safety, report any safety events, and monitor their labs. And of course, I can always be, if I'm offering drugs through expanded access, I have to generate an annual report, and I could be subject to any inspection or audits to ensure that I'm monitoring and maintaining safety of the person receiving the drugs, and I'm compliant. So there are a lot of extra steps that make it a little bit more challenging. As many of you know, there is legislation that passed in December, and this is Act for ALS, which allowed funding to kind of alleviate these theoretical hurdles. And the hope is that we are able to kind of overcome these things, but there are limitations. Not any drug could be eligible for this program, but and there are also limitations on what we would do with it. So I'll go over that in a little bit, but essentially the bill includes, or the legislation actually, I should say now, include grants for research, as you know, and as you've heard many times from many people, that we need to understand the disease better. So having basic science research supported in parallel to the expanded access programs is important. Trying to build a public-private partnership, and I think that's the only way to really understand and prioritize what's important from the community's perspective, and also trying to create an EAP granting program. But if you apply for this expanded access funding, you have to build in research goals. And so whether that's trying to find a biomarker or trying to validate outcomes or looking at survival or function, it has to answer a research question. So just yesterday, the NIH released a funding opportunity announcement with details about what's required for it. So in order to apply for this research funding opportunity, you have to, if you have a drug that you can potentially access through expanded access, you need a may proceed letter from the FDA, and it can't be an already approved drug. So through like a new drug application or a biological licensing application. It has to be an intermediate EAP, so it can't just be for one person. So it must treat a group of patients. And it should not and must not actually interfere with ongoing clinical trials. And there has to be an elaborate explanation as to how clinical trials will be safeguarded when offering expanded access. And again, there has to be a scientific question. So it allows access and it provides funding, which are great. We've come this far. And now it's about using or leveraging that access to learn more about ALS. So as asking questions about the biology of a particular drug, or developing a biomarker, understanding the biology of ALS, or developing clinical outcome measures. And then the last thing I think I'll just land on here is the challenges and opportunities. Many of you are familiar with a lot of these, but it seems to change a little bit each time. So obviously we all know the disease is heterogeneous. So that means that everybody's ALS is different. And everybody's disease progresses differently. And I think that's one of the things people ask me over and over again when they're newly diagnosed, and it's not really well understood. And the hard part is I can't predict yet who would be those individuals that progress faster or slower. And developing those markers to help kind of have that conversation would be helpful. One of the other challenges is that sometimes we're in such a rush to kind of speed the development of a drug and gain access that we don't really understand the behavior of the drug or the optimal dosing in preclinical or early phase studies. So sometimes, for example, if there's a drug that does something great in an animal model for ALS, or in stem cell dish models for ALS, the reports are covered and it gets into the media, and then people want to get access to it, but we sometimes don't know if it's safe to give in humans. We don't know what dose, and we don't know if it would be effective. And so it's really important to understand those before you enter human clinical trials. The more you understand there, the bigger the effect when it's time to provide it to people. And then, of course, because of these recent legislation on EAPs and Act for ALS, we have to know how to do clinical trials and implement expanded access without impeding the clinical trials. And I think as a community, we've come to terms on a couple of things, which we understand that clinical trials are there, and those that are not clinical trial eligible will hopefully have access to expanded access. Hopefully we can stagger the recruitment of clinical trials and offering expanded access so that they don't compete against each other. And trying to offer open label extensions to those that are in clinical trials so they have long-term access as well is important to kind of make sure clinical trials are safeguarded in addition to EAP. And then the newer challenge now is handling new drug approvals while trials are ongoing. I don't think I have an answer to that yet, but it is something that will pose challenges. So if something is in late phase clinical trial development, for example, phase 3, and now oral edaravone is approved, and plus or minus other things that might come down the pipeline, when the results come, it might be hard to tease out what the benefit of that investigational drug was if people are allowed treatment, but ethically, we should allow people to be on treatment, and how to handle that statistically, and also to determine efficacy is going to be very important going forward. And so how do we mitigate disease heterogeneity? We talked a lot about it earlier, so I don't have to belabor the point, but trying to develop better outcome measures, trying to find the sources of why people's disease are different. So whether it's a genetic mutation, or maybe how they presented, or maybe there's a marker. It would be great if I could have a marker, like if everybody came in and I could test the blood and I say, oh, your ALS is predominantly neuroinflammation, or yours is mostly oxidative stress, and kind of pick out which therapy would be great. I hope for that day to be a real one for me as a provider, because then I could decide, combination therapy, or maybe one may be better than the other as a first go-to or first line. Those would be great markers. And then of course, trying to understand prognostic models and patient selection. So maybe a drug has a particular target, and we know that this particular patient population has that abnormality. So if we line up the drug with that population of patients, we might get a bigger effect. And so this is kind of a graphic to show how historically we have just put everybody in one bucket and tried to use a single treatment, but we have mixed effects or mixed readouts. And so it's likely that not everybody's the same, and trying to be more thoughtful about which groups of people will respond better will make our clinical trials better. So that's definitely an opportunity for us. So for example, I'm just going to throw a couple of examples really quick. So a lot of people come to me and say, clinical trial eligibility, they're always looking for early people in early stage disease, and it's really hard and restrictive, and it often excludes a whole bunch of people. Is there any way to make the inclusion criteria or the eligibility criteria wider? And so it requires a couple of things. Number one, if you use a wider eligibility criteria, then you might be able to theoretically identify a subgroup that might respond, but allow for more people to be treated. And you would have to discuss with your regulatory authority on the acceptability of that cohort, and have it as a pre-specified group for your primary endpoint. But then you'll have the other people that are not in your primary cohort, and they'll provide additional safety and tolerability data, so that you can, when it comes time to getting it approved, it could be approved for a broader patient, broader group of people living with ALS. And then of course, this is my ideal infographic. I'm changing it constantly, but the other thing is that if people don't become eligible for the clinical trial, the hope is that expanded access would be offered to those that are screen failures that could provide safety and tolerability data as supportive data to the main clinical trial. And so that might be one way. And then if placebo has to be used, is there a way, especially if we're looking at rapid progressors or really rare mutations that are more aggressive? And we've employed this in the FUS clinical trial that's actively enrolling, looking at people, if they progress to a certain point, then transitioning them to the actual treatment, but they're blinded, but they actually have a chance to be on treatment. So that's a time to event. It's not traditional, but we've done it in ALS a long time ago, but we didn't kind of continue. I kind of wish we kind of developed that time to event or failure type of endpoint. The other is the other way. So I just talked about widening the inclusion criteria, but the other way, the flip way, is maybe making it more restrictive based on either the gene mutation or a biomarker or the mechanism of the drug. So we have got a group of people, and whatever drug we have targets a specific biomarker or gene mutation. So we have a smaller group, and the hope is that when we do a precision or targeted approach, we'll have a bigger effect. So we don't need a large number of people. And then we already talked about this, but Silence ALS is another way of foundation and university and academics all partnering together to kind of figure out very personalized precision type therapy. So this does require not only collecting samples and evaluating the underlying tissue and blood markers, but it also does take time to, in rare mutations, it takes time to apply what we call antisense oligonucleotide technology and tweak it for the particular mutation. So that does require some time to do toxicology studies, and you can't just jump right into people right away. So, but it is a way to be more precise on targeted therapy. And of course, being more efficient, many of you are familiar already with the platform trial. So as many of you know, regimen A, which was the LUCA plan, underwent an interim analysis and it was stopped for futility. So that was regimen A. That was just a couple months ago. And then regimen B, C, D continue, and regimen E is recruiting. And so when regimen A was stopped for futility, it met its goal, which was really the point of this design. It's not, if there are many drugs that people want to test in ALS, trying to be efficient in the number of people and whether we need to continue development and spend resources and have people on that therapy, trying to shorten those times of exposure, trying to get to an answer quicker. That's the whole point of this. So it's hoped that the way this adaptive trial is set up is that we can kind of answer these questions with less number of people, less cost, and terminate if we need to, or graduate it and move along and move it for approval if it shows effect. And then we talked a little bit about this in our conversations earlier today, but prediction algorithm, I only have this is from Origin Data Sciences because I've worked with them closely on developing some of the aspects of their predictive algorithms, but there are algorithms out from NCALS, which is very good and user-friendly, and also Patients Like Me, and there's several others that are developing AI specifically for use in clinical trials or outcomes or cohort identifications. But the way you can apply these in clinical trials is, as we talked about earlier, is control arms. So if you have a phase 1 and you want to treat everybody and create virtual controls, it might give you some early signal to move forward. Sometimes you can actually make sure that you have fast progressors and slow progressors equally distributed across treatment and control arms so that you don't have to use such a big sample size and it increased your power. More recently, they've been working on cluster analysis or looking at hotspots. So you might have a clinical trial, might not have worked, but then you can do cluster analysis to look at areas where you had good response and kind of identify maybe your responder groups that way. And that's just one example of using predictive algorithms or AI. Another challenge, like I mentioned, is incomplete understanding of the drug. So in the lab, in the preclinical phase, usually there's efficacy testing in the models for ALS, and you optimize the compound, and then you test it, and you do toxicity testing, and then you manufacture it and scale up so that you can start your human clinical trials. And usually regulatory agencies have a package that you have to meet in order to get clearance to move into human clinical trials. But once you get there, sometimes there's such a rush to get to the late phase clinical trials that some aspects are skipped so that you don't get full pharmacokinetic information. So you don't get a full understanding of how the drug behaves and how it goes in and out of the system. You might not understand what the ideal or optimal dose was. You might not have understood whether the drug actually engages the hypothesized target. And you might not understand what the optimal dose is. And so when you skip those phases, you're at higher likelihood of failing. And so it's really important. And, actually just earlier today, I was in a meeting where we were talking about what would we want as a scientific community from people in the preclinical phase, like what animal models, what models do they have to show us? Do they need that for FDA progression from preclinical to human clinical studies to get an IND? And I think we know that there's information that's required for safety and toxicity testing and for dosing, but ALS specific, what do we need to have confidence that it would have an effect? And that's not an easy question to answer. So that's something I think the community has to come together with and kind of put forth. And the last thing I'll leave you with is the resources for clinical trials. So of course, we had a discussion about how it was hard to find information about clinical trials. I am the NEALS co-chair, so you're seeing the NEALS logo and the NEALS database, but I'll also show you, this is one way to search for a clinical trial, and there's a support person, Carly Allen, who can talk to you about questions if you're looking for ways to participate in clinical trials. And there's a search tool within the website. I Am ALS also has a website called Als Signal. This is actually for people who like to be on a site and change things. It's actually pretty cool. But you can actually look for a clinical trial and tweak it to the region, the phase and whatnot. And they also have EAP information on their website. But if any of you have gone, and clinicaltrials.gov is the other website. If any of you have gone on any of these websites and done a search, so I know, Terry, you were looking for my presentation, I was still checking on clinicaltrials.gov like the minute before I started talking today, to see the updates on the drugs, and it's cumbersome. And so first you have to know that the databases exist. Second, you got to do the searches and know which keywords, and it becomes hard. And so my hope is that, and this is my own personal feeling, my hope is that there's some way that we can get people matched up with clinical trials as soon as they get diagnosed. And that's like thinking way out of the box. But in oncology, you know, I had a recent family member with breast cancer, and you know, the tumor was taken out, it was sent out, they did profiling of the protein expression, you know, and I was like amazed at how sophisticated it was. They looked at the biomarkers and then decided which hormones were affected. And then she had some therapies that she would try, but if that didn't work, then the clinical trials. So in oncology, they already have a system, I have several systems actually, where a patient is screened, right? So once the tumor is taken out, it's screened, that automatically links to a database that searches all the clinical trials and matches them to their unique biomarker feature. And then a report goes out to the physician who actually looked for the biopsy or who ordered the biopsy, and they'll get a list of the clinical trials, so that it's not, the onus is not on the person living with the disease who's going through so many things to go search out a clinical trial database and find the information and figure out where to go. So I'm hoping, and that's a big thing to do in ALS, but I think it's possible. And I know that there are many groups that are looking into this matchmaking tool, including the ALS Association. And the other thing, because ALS clinical trials, there's a big pipeline, and they're often looking for people who are early in their disease, but often people will show up on average one year into their symptom onset, and a lot of people will show up even after that. And so they are not eligible for a clinical trial because they are too far away from when they first had symptoms. So the other is educating our own peer community to identify and think about ALS as early as possible. So this is a tool developed by the ALS Association, and it's meant for physicians outside of the ALS community. So general neuromuscular clinicians, neurologists, primary care physicians in the US, so that if there is evidence through this kind of algorithm of ALS, they make a quick referral to the ALS center, and hopefully that could start the process of diagnosis, confirmation, genetic testing, multidisciplinary care, and review of FDA treatment, and then hopefully provide more opportunities for clinical trial eligibility. So again, speed, innovation, access, we've heard it all. I put the use of real world evidence here as a potential and expedited pathways for post-approval data. I think there are mechanisms for this. In ALS, we're limited because we don't have a surrogate marker that's been validated from a regulatory perspective to meet the criteria for accelerated approval. So we have to meet full approval requirements, which is usually 2 phase 3 studies. But if a phase 3 study or a phase 2 study, say you're in phase 2, and it shows a benefit in function or survival, is it okay to do another phase 3 and just hold off? Or should we provide access to those living with the disease now and consider a way to collect data post either full approval or do a confirmatory study while a drug is on market? I mean, there's ways to do this without compromising standards of regulatory evaluation and scientific evaluation, and other diseases have done this. I didn't think I put that slide in there, but I'll throw it in there. So I made this slide for myself actually, as the landscape is evolving and there's drugs under regulatory review, and we have the ALS drug development guidance, and we know what the standard is for full approval. You usually need two confirmatory phase 3 studies, and that's typical for every other disease. But can we establish a standard that's relative to the unmet need of ALS? So of course, when we look at a clinical trial, we want to look at efficacy and the magnitude of effect and what is it affecting. You'll have a statistics talk later that will kind of emphasize some of these statistical issues, but looking at the data of a clinical trial, and was it pre-specified, and was that what they planned, or did they dig through the data to find those responses, which will sometimes be fraught with chances of being just a random finding. Statistical evaluation, was it safe, and what were the serious adverse events and what were the common adverse events? Did people drop out because they couldn't tolerate the drug? Is it easy to take, and how does it fit into the current treatment landscape? And how can we collect data if we get something out there early and it hasn't completed a second phase 2, what can we do as a community to meet either the requirements for accelerated approval, so that's developing a biomarker or a surrogate, and validating, or how can we collect data to have confidence in the efficacy of a drug? And those are not answered questions, but I think as a community, we have to kind of work really hard right now to come together as one, as was said before, we're all working together. And I think it's important for us to come together as a community to really answer those questions, and we can make it easier. This is an opportunity to make clinical trials more effective going forward. So again, bottom line, we still need basic research. The better we understand what's causing your ALS, the better the targeted therapy would be to make a bigger impact. That's the bottom line. But we also have to facilitate translation of genetic and cellular therapies into clinic. And sometimes the traditional phase 1 to 3 doesn't quite work for those types of therapies, but we have to understand how to modify that drug development timeline to fit those particular therapies. It should be mandatory that every clinical trial has some sort of biomarker that we're collecting, even if it's experimental, so that we can build repositories and grow them. Those require funding to sustain the infrastructure and fund the people that run it. It sounds so easy, but it's so complicated, as other people could tell you. Clinical trials are still the most important way to determine true efficacy, but expanded access could help to provide additional supportive data for those that are not clinical trial eligible. And of course, we have to continue trying to innovate in clinical trial designs like adaptive trials, platform trials, patient stratifications. I really want to push on the use of AI and predictive algorithms to see if there's ways to develop efficiencies that way, or look even at potential groups or cohorts that might have been missed and we didn't see it in the big picture, but maybe looking at the data that way could help us find a new kind of group to look at for a future clinical trial and developing better endpoints. So there's a lot of opportunities, a lot of challenges, but a lot of opportunities and a lot of work ahead of us. But I think that's these are all things that are doable, but we got to think a little bit outside the box to try to achieve these goals and be brave. And with that, I will conclude and take any questions.
Thank you so much, Jinsy. That was great. And it's a real privilege to have you here. So we do have a couple of questions, I think, or comments in the chat box that Pat asked, what did you say about accelerated approval? Why does ALS not fit that approval?
Yeah, that's a great question. So if you take, I just laughed a little bit because some comments about my knee, but the accelerated approval in its formal definition requires, so in order to meet that, a study has to evaluate a drug and look at an outcome, and that outcome has to be predictive of whatever the gold standard is, which is survival in ALS, actually. And so that marker or that outcome is called a surrogate marker for whatever the gold standard is. And in order to be defined as a surrogate marker, you have to get validated or provide a lot of data to the FDA to prove that it is indeed a surrogate. Now, ALS functional rating scale could theoretically fit that. And that's why there's kind of this gray area and why we're struggling a little bit when we have a study that has an effect on ALS functional rating scale, and it might be 2 points, it might be 2.5 points different, and it's still function and it's still important. And so, but it's not considered a surrogate, right? And in typical, if it wasn't ALS, that type of data would require a second phase 3. And so that's when I think there's a gray area, and our community, I don't think, has come to understand where we sit and on the level of evidence for approval. If, like, for example, neurofilament was predictive of survival and predictive of ALS functional rating scale, which we don't have that evidence. We think it's there, but that evidence is not quite clear. Neurofilament is still a great biomarker. And so if it did definitively predict clinical outcome, that would be one way we could kind of achieve accelerated approval. The drug development guidance that was finalized in 2019 provides some context relative to ALS. That was the intent of that document. And so I think it's important for us to look at that and see if there's ways to move forward. So it's not that I don't think accelerated approval can't be applied, and there is. So just to be transparent, I think there is a concerted effort in our government and also with our regulatory agencies to figure out how to improve on accelerated pathway in the US. And I think they're doing it with the intent of trying to make it easier for groups like us in the ALS community when we see results like we do in clinical trials.
We have another comment from Kevin who says the concept that clinicians would look for or recommend trials for individuals would be welcomed by the whole PALS community. And I do think, and I don't know how the other clinicians on the Zoom feel. I know that I always sort of educate folks about what trials are actively recruiting and even provide something in their after visit summary on our EPIC system and especially explain why I happen to do genetic testing on everybody because of those trials. So I open the floor up to anybody else to comment for Kevin.
Yeah, go ahead, Lau.
I would just say, I think our job as clinicians is to make sure that our patients, people with ALS are aware of the trials that they would qualify for and help them, enroll in those trials. I would be very cautious of a provider telling you that they think one trial is better than another, because look, for the past 40 years, we've done a crappy job of predicting that, right? And so anyone who tells you, oh, you should do this trial instead of that one because this is a better drug, forget it. We don't know what we're talking about, okay? If you want to be involved in clinical trials, and you definitely should be involved in clinical trials, and we'd love to talk to you about why that is, choose one based on but we're going to tell you what the risks are. We're going to tell you, maybe you don't want something that's injected. Maybe you want something where you don't have to travel as far from your home. I mean, there's lots of reasons to choose. The fact is that every trial has a limited number of spots. And as you know, you might only qualify for a trial in a particular phase of your disease. If one's offered to you, take it. And I wouldn't worry too much about ALS physicians saying one trial is better than another, right? Because we're not a good judge of that. So, that's what I would say.
Yeah, I think I would add also the multidisciplinary style of care often is a lengthy visit. And we're trying to do a lot in one visit. And then I will try to squeeze a clinical trial discussion in, but it's difficult if someone has been there for 2 or 3 hours already. And I think that sometimes creates a problem with trying to have that discussion. And so I've heard that from other people, and I'd really make it a point to connect with people that I see personally and talk about what they might be eligible. So sometimes it's not even at my center or not even in my region. But one of the things that I thought about, because it's important, so I talked about the Think ALS tool, but there's many that have talked about, well, why don't we say Think ALS trials and make it part of our practice to talk about clinical trials? And I was like, well, we do a lot in the clinic, and we have to find a way to facilitate that, and not add more burden to people who are already coming to those clinics and the people who are providing the care. So one of the things that I thought could be helpful, and some places already use this, is like the research navigator or a research, like a research navigator, research access person, and other specialties do this, but some centers have resources and are able to pay staff to specifically talk about clinical trials separate from their visit, and they can do it as a Zoom call. But, I've pitched to some groups, foundations, to see if they would fund like regional access navigators. So like, there's a whole bunch of, where I am in New York, there's a whole bunch of us ALS centers. But if we had one person who knew what all the sites were doing and do that extra visit as a research navigator for the tri-state area, for example, that would alleviate my burden for sure. And so that, I think if there was a network of like regional research access navigators and we had funding for it, it's expensive actually. So I think it was like 15, some of you were on that call. I don't know. I think it was like, if you did 15, it cost like $5 million or something for 1 year worth of funding, but even a couple will help, So,
one other thing I would point out, so we really do believe that one of the problems is that perhaps we need to target the therapies. If a therapy's working on a specific mechanism, then maybe we need to figure out who are the patients that therapy is going to work best in. But we may be wrong about that, right? I mean, pharma companies considered the most de-risked trials these recent genetic therapies, right? So, you know, a predictive biomarker of a therapy that targets an SOD1 mutation should be the gene test for SOD. And as you guys know, you know, the recent trials, at least with SOD and with C9, you know, these results were not what we were hoping they would be. Now, that doesn't mean they're not going to be better in some of these other therapies that are, you know, currently being developed. But time will tell. And so all we can do is the best we can do in trying to match patients to the best trials. But honestly, if you are on board to be in a clinical trial and one's offered to you, take it. I mean, don't worry too much about what the trial is. Go for it, right? And part of our job, and when Kevin was asking earlier about what can we do to accelerate this, like we're going to talk about this tomorrow, but I want to make sure that the right therapies are getting to the freaking platform trial, right? I don't want the platform trial to be enrolling something because some pharma company has money to spend and they say, oh, we'll pay you to do our drug, right? I want the best stuff that's the most, you know, promising stuff to have the biomarker data, to have the tox and the manufacturing that Jinsy was just talking about. You know, we need to make it so that there is a menu of things for the platform trial to carry forward. And that's how we can speed this up.
Yeah, I agree with everything that was said, especially I think what Jinsy was mentioning, that, you know, we'd like to have a profile getting, you know, what is the most likely target in an individual patient. I mean, you probably have read and are aware that there are certain, you know, centers like the New York Genome Center has this big project where they analyzed data from postmortem tissue, and they came up with this, you know, 3 different major group of potential, you know, signatures of disease. And so the challenge is really not waiting that, you know, and having that information postmortem. We wanted at the very beginning when we first diagnosed people and kind of attacking this, you know, signature intensely so that we hopefully, you know, reverse some of the disease. That will be, first of all, stop and then reverse.
So thank you.
And I also,
so go ahead. I just want to quickly say, like, so Jinsy mentioned earlier, she wants to be able to do that, right? She wants to be able to test somebody when they come in and say, which cluster are they? So Selena was just mentioning this project. So I'm the one who, you know, organized that project, right? And so you're right. We identified these clusters in the brain tissues. And so I mentioned quickly, you know, that we're looking at muscle now. One of the things we're excited about is the idea that perhaps we can find those same clusters in muscle, right? Because while I can't biopsy your brain during life, I can biopsy your muscle. And if we can find things that identify those same 3 clusters, you know, in a muscle biopsy, that could potentially provide what Jinsy was asking about and help us decide who goes to what therapy. So we'll see.
so, and I would also like to add something that, Jinsy, you said something very, very important, that we also have to consider the incomplete understanding of the drug, right? In particular, the pharmacodynamics and pharmacokinetics, as well as taking real world evidence. And I would really like to end here with your first slide, showing the effects of riluzole and a little bit of optimism, I think, because we keep, now we have the tools, I think, and we have shown it. That riluzole, actually, if you look at the first slide that you showed, and you look at the effect of riluzole in the first year of treatment, when you start treatment early, riluzole is indeed effective in slowing down disease progression and eventually prolonged survival by 86%, which is a lot. As year 2 starts, boom, the effect goes down to 12%. What we have shown, and then we have done the proof of principle in the mice, starting treatment in the SOD1 mice at onset, okay? And that has never been done. And that's why I don't think that the mice are not predictive. It's just that we didn't use the mice as we should have used them. But that's a different conversation. So what we have shown is that also in patients, starting with the mice, but also in patients, as disease progresses, there is a disease-driven increase in pharmacoresistance that is not present in control and is not present in early symptomatic patients, and therefore that starts kicking out, pumping out riluzole and any other drug from the brain, that gives riluzole not the effect that is our desire. So here we have an opportunity also to revisit some of the clinical trials, and in particular also the treatment of riluzole, by grouping really patients in drug responsive versus drug resistant, and combine the use of riluzole and any other trial with perhaps some blockers of these PAPs. Sort of what they've done in HIV. And I would really like to end a little bit with the more optimism with all the challenges that we all presented, all the challenges that ALS gives us. But here we now have a community of researchers, clinicians, clinical trialists a la Jinsy, people from industry who have done it like Jinsy has done it before going back to academia and to treat patients, that study pharmacodynamics, study pharmacokinetics, and improve the drugs that are already available, or while we are testing them in trials, try to improve them by that, and really look at the real world evidence and go back to try to improve what we already have available, I think would be a fantastic starting point. And that works for every patient, right? While we are trying to do the stratification, while we're trying to do the personalized medicine. But that's, you know, it's 7 o'clock in the evening, and I wanted really to end with a little bit more of,
Rizol works, and it works better if you give it early. And, you know, I often say to patients, if I think they have ALS, I say, look, I might be wrong, and I hope I'm wrong, but let's start Rizol, because 6 months from now, it's really easy for me to stop a drug. It's really hard for me to say, shit, I wish I started that drug on you 6 months ago because I don't have a time machine. And so, you know, yeah, Rizol slows the disease down, and if given early, does it better. And it's remarkable because it does it for just about every flavor of ALS, right? So.
And now we'll have an oral version of edaravone. So, you know, which reduces the downside of edaravone, which is the IV and its impact on quality.
And Terry, I'll tell you, one of the things I'm attracted to about that is also the IV form of edaravone, the way that it's on and off, screws up all our biorepositories, right? Because suddenly we have to pay attention to when somebody's on or off drug if we're collecting their blood or whatever it is, because it affects like all these markers in the blood. So, you know, anyway, hopefully it'll be the drug. Yeah.
So there's a question from Joe Lustro, or comment. It depends if we want to take it now or you want to discuss it tomorrow, Terry.
Let's see. Oh my. This is interesting, Joe, because I've actually been concerned because neuroinflammation probably occurs in the context of COVID and does drive disease. And I've had several folks who insist that their disease accelerated and or began when they got either the vaccination or they got COVID. There's no way to prove it because it's, you know, at this point, we need to collect the data, but it's an interesting concern. And I don't know how others feel about it. Rick is talking about, he published a couple of papers on this. And I think I actually talked to you about this, Rick, because, you know, I think that the mechanism is neuroinflammation. So Rick, maybe you want to comment on this.
Yeah, Joe, I've seen the same thing in my patients. So we published 2 papers on this last year. I'm happy to send you the references if you want them, just email me. But one was a couple of cases of people that I had followed for years who were hardly progressing. They were doing extremely well. And in both cases, when they got COVID, their acceleration just exploded. One actually died in a couple months from ALS progression. And the other one, you know, went from walking, he'd been walking for like 6 or 7 years to wheelchair bound just a few months after getting COVID. And then the other was actually a review of a fair number of cases in a VA database looking at survival after COVID. And it appeared if you had an ALS diagnosis and you got COVID, you were very likely to die in the 30 days after the infection. So I really do believe there's a connection. There's somebody who's doing a much better, more scientific review of this. So Hervé Desai at the Carolinas ALS Clinic has a COVID registry. So if you Google Carolinas ALS Clinic COVID registry, he will want to hear about your story. He will want to, you know, have you answer some questions and probably get some medical records on you because he's trying to put together a much larger number of cases.
Okay, thank you. As proof, I was that I was paying attention, I understood the correlation is not causation.
Yeah.
So that's sort of.
There's, the other thing that we always come, now maybe not with this COVID story, but it's not uncommon for us to hear that somebody's ALS symptoms started after a surgery or after an injury or after an illness. And the one thing we always wonder is, are those things triggers, or is it possible that somebody just, you have to lose a certain amount of your motor nerves or your muscle cells before you actually notice that you're weak. And when something is progressing gradually and slowly, it may be what we call subclinical. And it may just be that you don't have the functional reserves to deal with another insult. For example, if I get sick, I don't have ALS, right? But if I get sick and I lie in bed for 4 days, and then after those 4 days, I get up to go, get back to my life, I'm weak and I'm exhausted and I have trouble getting through my day and my muscles are all weak. And so if I perhaps had mild, subclinical loss of my strength up to that, it may just be that being ill or having hip surgery or a dental procedure, whatever it is, if I'm laid up for some other reason or ill for some other metabolic or infectious reason, I may just not have the functional reserves to deal with that. And so then I become symptomatic because those two things combine. Now that's a little different than what Rick is describing, which is that, and it's, I think, coming out more and more that in this case of COVID, it appears that there really is perhaps something that is accelerating disease. So people who, are getting COVID and then something is going, nuts in their system. And maybe that's not surprising because, as we know, again, these general sorts of mechanisms, a lot of them are immune mediated or inflammation or, the kinds of things that you would expect this to affect. There's also an interesting biochemical part, which is that the way that COVID gets into your cells is through what's called an ACE2 receptor. And the ACE2 receptors, there's very little data in humans, and nobody's really looked in postmortem tissues in humans, but it's well described that those receptors in mice are on spinal motor neurons. And so, it's sort of not surprising to me that something's falling out of this because the receptors that virus uses to get into our cell are on the cells that are involved in ALS, at least in mice.
Thank you.
But it will first has to pass the blood-brain or the spinal cord barrier, right?
Maybe only antibodies do, right? I don't know.
Okay.
Maybe the blood-brain barrier is disrupted in ALS patients, and certainly Pierre is one who's that sort of thing, quite extensively, and how it's remodeled. So I don't know. We'll figure it out.
Yeah.
So I want to, thank Jinsy for a great talk and great discussion. And that actually completes our sort of coursework for the day. And right now we have an opportunity. We have another question, Jinsy. This one will be good for you. How long does it typically take for a newly approved drug to get to the market? Variable. Phil's asking that, but why don't you go?
It's variable. And there is some, with the oral edaravone issue, they had gone through the IV approval process. There was bumps along the way in trying to get access, and I put that gently. But I think that they've learned to do some work ahead of time in the anticipation of an approval. So hopefully that won't be so, meaning drug manufacturing in the US, because there was no drug manufactured in the US at the time of the intravenous formulation, working and trying to figure out formulary placement and so on. Typically, it can be anywhere from 6 to 9 months. I'll just tell you, in the ALS community, post edaravone approval, I don't know what it was like for everybody else, but the approval came May 2017. I think I dosed my first person in January 2018 because there were so many hurdles in trying to get prior approvals, denials, people, companies not understanding it. So it was difficult. So sometimes that can be there. But I think every manufacturer that is heading towards phase 3 pivotal phase are I think are mindful of the fact that if something should get approved, they should do the work ahead of time. So once you're in phase 3, companies in the ALS space, I'm speaking from my own personal experience, will work with regulatory agencies in the regions they're expecting to file for an approval and do the work ahead of time to minimize that time. But there's always a little lag time depending on whether that region has a drug depot and has manufactured drug in the region that's available, how fast it can be reviewed by the payer system and can be reviewed and made a determination so that it allows access. So I think everybody wants to gain access if something is effective.
Jinsy, one last very fast question. So was it very clear from the Japanese, the methylcobalamin study, that they have actually slow disease by much more than 2.49 points, which is the Radicava slowing. I mean, if I kind of read the slide correctly, that was from -2 point something to -4.
Yeah, the difference comes out if you do the least mean square, it comes out to 1.94 difference, which is a little bit less, but also,
Oh, okay.
but it's also in a shorter duration of time. Also, in the style of Radicava, the population were people who were early, very, very early stage, rapidly progressing. So it's a very homogeneous population. So that's why they were able to do it in a small number. And so again, I think there'll be questions, but this is again where you talk about, okay, B12, how methylcobalamin, what is this mechanism? Is it complementary to what's already available? What's the risks, involved? And there are financial risks, right? If you have to pay for something, that's a risk. What are the side effects, and all of those things have to come into consideration. So.
And Rich is adding that there's no benefit if it's after 1 year of symptom onset.
Yeah, for this clinical trial, for this particular study. And that is this uncertainty and this kind of unclear, interpretation. If you see this, then how does it apply to everybody? And that's, I think, what we're contending with right now as a community.
Yeah, and it would probably imply that the mechanism by which it works is one of the inciting mechanisms rather than the propagating mechanism.
Right.
So, but well, this has been really a great discussion. And again, I thank Jinsy and our faculty for jumping in and commenting, and for all of our participants for really making this an active discussion.